Welcome to this introduction to the use of proteomics in colloid and surface biology. I'm Cristina Furlan, and I'm a lecturer at the System and Synthetic Biology Department at Wageningen University and Research. How do you feel about drinking water coming out from this pipe? This pipe is covered in a biofilm of microorganisms that interact among themselves and the surface material of the pipe. How does this happen? Major effectors of these interactions are proteins. To study the plethora of proteins involved, we resort to proteomics. Proteomics uses mass spectrometers in combination with biochemical or biophysical methods to identify and quantify proteins. At the end of this video, you should know what mass spectrometry-based proteomics is and how we can dissect the factors of interaction between cells in a biofilm using proteomics. Proteins are made out of amino acids bound together in a chain. We try to find out information about the identity of proteins with mass spectrometers by looking at smaller parts of their sequence, their peptides. This approach is called bottom-up or shotgun proteomics because we look at all proteins present at a given time through the analysis of their peptides. To obtain peptides from proteins, we use enzymes like trypsin. Trypsin is the same enzyme that digests proteins in your smaller intestine in real life. How does a mass spectrometer help us in characterizing the peptides of a protein? A mass spectrometer is an analytical instrument that allows us to measure charged species. It is basically a scale for the peptides. When our peptides have passed through the mass spectrometers, the results are visualized as a graph in which the x-axis shows the m over z, meaning the mass over charge of a peptide, and the y-axis its intensity. This graph is called mass spectrum or MS1 spectrum because the peptides went through one round of mass spectrometry analysis. Yet, we want information on the sequence of the peptides. And we obtain that by breaking peptides up into smaller pieces in the mass spectrometers. Basically, after the MS1, specific M over Z are selected for fragmentation. Some energy is provided using different methods, so to break the bonds between the atoms. The mass of the resulting fragments is analyzed and detected. The return spectrum is called MS-MS, or MS to the second spectrum. The peaks in the spectrum correspond to fragments of the peptides. Finally, via bioinformatics analysis, we can derive the identity of the peptides first and build back those into protein levels identifications. This is a well-established bioinformatics pipeline when we have genomic information for the organism investigated. Now, we have seen how we can identify proteins present in a sample, yet which samples are we interested to study to describe cell-cell and cell surface interactions? Let's go back to the example of our biofilm and move from the inside to the outside of our community. We can look at proteins that are present within the bacterial cell or at specific organelles or part of the cells. We can also look at proteins that are at the surface of our bacterial cells. And finally, at proteins that are secreted. These three different levels require individual approaches to select the proteins of interest. Let's start with the cellular or intracellular proteome. First, the extraction of proteins from the inside of bacterial cells can be done via many methods. Lysis buffers of Weber might affect what type of proteins you extract and therefore what you see. This problem is highlighted here in the figure via the different amount of ribosomal and membrane proteins extracted by using different buffers. Another important parameter to consider is the amount of starting material. Samples from natural environments might not have a very high cell number. To solve this, it helps to perform the steps after extraction on a solid support. In this paper, for example, they use sp3 beads, improving considerably the number of proteins obtained from 2 million cells. Now, let's move to the surface of the cells and look at what is called the surfacosome, or surfome. By looking at this subproteome, we can obtain information on proteins involved in adhesion of the cells to the biotic and inert surfaces, and proteins that mediate cell-to-cell interactions. There are many methods to select for surface proteins, the easiest of them being shaving the cell surface with proteases, such as proteinase K and trypsin. Trypsin being the same enzyme we use normally in digesting proteins in a proteomics pipeline. This figure summarizes all methods available, but the take-home message is that none of them will give you a complete picture, but using a combination might be more advisable. Finally, we can look at the subproteome containing the soluble proteins, also called secretome or exoproteome. These proteins are involved, for example, in cell wall metabolism, degradation and uptake of nutrients, or binding to substrate or host, and are important for the establishment of the biofilm. Together with proteins and other biomolecules, there are also extracellular vesicles in this environment. There is a basic method to obtain a secretome sample for proteomics analysis. This involves centrifugations or ultracentrifugations to remove cellular component or extracellular vesicles, followed by a precipitation step via acid or organic solvent, or in alternative, a concentration step via spin column with low KD cutoff. All methods for sample preparation need to be complemented with bioinformatics analysis involving localization prediction tools. These disentangle true results from contamination to the extraction methods. To sum up, we have seen how we can use mass spectrometers to retrieve information about proteins by analyzing their peptides. Basically, mass spectrometers are scales for charged peptides. Their measurements for the charged peptides are stored in mass spectra. A mass spectrum reports the weight over the charge and intensity for the peptides, or fragment peptides in case of a mass spectrum. Via bioinformatic analysis, we can then retrieve the identity of the proteins. We then have investigated together the 3 types of subproteomes relevant to colloid and surface biology. Those subproteomes include cellular, surface, and secreted proteins. We have also seen some methods to obtain those subproteomes and factors that might affect their preparation. If you wish to know more, please check our extended seminar linked in the resources. But now I won't keep you further. Go and start analyzing your favorite sample by proteomics and uncover the power of proteins.